So children, today we will discuss about the transformation method. Now what is actually transformation? Transformation means that the transfer of the rDNA into the host cell. Now in this case, host cell, it can be bacteria, it can be plant cell or animal cells. Now let's discuss in detail. As all of you know that how the rDNA that is constructed, rDNA, it means that the foreign gene, the foreign DNA with having the plasmid DNA or the another cloning DNA in which the foreign DNA that is linked so that the multiple copies that are produced. But how these multiple copies that are produced when these rDNA that are transformed into the host cell. So today we will discuss some transformation methods like calcium treatment method, second that is micro injection, and third that is the gene gun method. So first that is calcium treatment method. So why it is required? It is required to make the host cells competent for taking the DNA. Why it is necessary? Because DNA that is hydrophilic in nature and it is not able to pass, it cannot pass through the cell membrane. So that's why this cell means the host cells should be competent. So here this method, it tells that how the bacterial cells they can be made competent by using the calcium treatment method. So the first is that the host cells they should be treated with calcium so that it create pores in the cell wall. Now next is that after creating the pores, means when the pores that are present over the cell, then incubation period should be there, incubating the host cells with rDNA over the ice. Then next step that is giving the 40 degrees Celsius electric shock and then again putting the cells on the ice, means again the cells as well as rDNA over the ice. Then it enables the bacteria to take the rDNA. rDNA means recombinant DNA. So basically what is the principle here to give the calcium treatment is to create pores over the host cells so that the bacterial cells they will be able to take up the DNA. And this process it will become fast by the incubation or the electric shock treatment. Now second that is the micro injection method. In case of the micro injection method, it is used for transforming the rDNA. Okay, transformation of the rDNA into the animal cells. Animal cells they contain the nucleus, as all the cells they contain the nucleus, but basically it is used for animal cells. And recombinant DNA through the micropipettes. Okay, micropipettes that are used for the transformation of the recombinant DNA directly into the nucleus, and this process is known as micro injection, as the recombinant DNA directly introduced in the nucleus through the micropipette. Why it is known as micropipette? Because it can transform the micro-sized particles in the cell. Now next we will discuss about the Biolithic method. Biolithic method that is used for the transformation of the rDNA into the plant cells. So now the third method that is the biolithics, and they are also known as gene gun, and they are responsible for transformation of the rDNA, recombinant DNA, into the plant cells. So here this diagram, it shows one tube. This is accelerated tube, and from one end of this tube the helium with high pressure that is passed. Okay, and here you can see the ruptured disc that is present, and next that is a macrocarrier to which the DNA coated microparticles of gold and tungsten that are attached. Okay, and here this is known as the stopping screen, and here the plant cells or tissues that are present. So where the rDNA that are present, rDNA that are present in the microparticles. These DNA that are rDNA which are coated with gold or tungsten. Now from one end helium with high pressure that is passed. So when the helium that is passed, then what happens? This disc, it will get ruptured, and then the helium with high velocity, it fall over the macrocarrier. So when it fall over the macrocarrier, then this macrocarrier, it is stopped by the stopping screen. Means it is blocked by the stopping screen, but these DNA coated microparticles, they pass through these holes and they will embedded in the plant cells, means they are able to enter in the plant cells with high velocity. Okay, let's see again. Helium that is passed with high pressure, then this rupture the disc, and then it fall over the macrocarrier, means macrocarrier it is stopped by the stopping screen, but because of this helium pressure, the DNA coated microparticles, they will pass through these gap which are present in the stopping screen. Okay, so they pass through these gap and they will be able to enter in the plant cell or tissues. So in this manner the transformation it occurs. Now next method that we have already covered, that is the disarmed pathogens. As the pathogens, they used to infect the plant cells and the animal cells. So if suppose the disease causing genes that are removed and they are replaced by the foreign gene, so they act as the disarmed vector for the transformation of the rDNA into the host cells.